Sourdough. Known for its signature tangy flavor and its intense multi-day preparation. Despite the complexity, it all starts with two simple ingredients: flour and water. This is a starter. The starter undergoes a fermentation reaction for a minimum of 6 days. It is fed more flour and water each day until it has fermented enough to make bread. The starter is actually alive. Let's take a closer look. Starch is a polysaccharide, or chain of sugars, that is found in flour. Starch is converted to maltose, a disaccharide, by an enzyme called amylase. Maltose is then converted to single sugar glucose by maltase, an enzyme produced by yeast. Yeast from the environment, called Saccharomyces exiguus, find their way into the starter. This yeast eats the glucose in the flour and produces ethanol and carbon dioxide, which makes the starter rise. Bacteria found in the starter, lactobacillus, digests the ethanol to make lactic acid. This lactic acid is what gives sourdough its signature tang. The starter is mixed with some flour, water, and salt before a 1-hour proof. The dough is then shaped for an 8-hour proof overnight. During the proofing periods, the dough is kept warm and covered. The purpose of proofing the bread is to allow for the yeast to further ferment the dough and create leavening. Fermentation is controlled by salt that draws water out of the yeast, slowing their ability to ferment the dough. Next, the dough is kneaded. Proteins, glutenin and gliadin, are found in the flour. Through kneading, they are manipulated to form chains of amino acids. They line and form an organized matrix. Salt helps to tighten this structure, which will play a big role in rising while baking. After 1 more hour of rest in the fridge, the dough goes into the oven. Typically, sourdough is placed inside of a Dutch oven for baking. During baking, the bread continues to rise as steam and carbon dioxide fill the pockets of the gluten matrix. Eventually, the yeast are killed from the heat of the oven, and the protein structure denatures and becomes stiff. The formation of the crust, or browning of the bread, is called the Maillard reaction. In this reaction, amino acids and glucose in the bread interact. More specifically, the carbonyl group of the sugar and nucleophilic group of the amino acid are interacting. After all this, the bread is taken out of the oven to cool. And then,